מי מטפל בזה? אפשר לכבות את המזגן? רוצים שאני אעשה? מה? לא יודע, תעשו הצבעה. מה? דיקטטורה חופשי, אני לא מנסה לטור לכם. Alright, so few things before we continue. So it turns out that we have 24 people, which is way more than I expected. And it required some changes in the schedule. So what I did, first of all, because it took some time to organize a reading list or the presentation, so today we had to have a session. I postponed it to next week. But so what I'm going to do today is finishing a little bit more stuff about neural coding, single neuron stuff, and then we'll switch to somatosensation. So today and Thursday will be somatosensory system, and in the second part of the somatosensory system I will actually do also the cortical microcircuit that was supposed to be a lecture on its own. And then we'll have a marathon of 4 sessions in a row of papers presentation. Now this is going to be a little bit tough because we have to also have at least 3 people in every time. And originally the plan was that you would read all the papers for all the sessions, and this means that you're going to read 12 papers in the next 2 weeks, which is a little bit challenging. So what we will do is that we'll decide in advance on which papers we're going to read together, so you would read those papers and of course the ones that you're going to present. So I uploaded the up-to-date list of the presentations on the Moodle. It's an Excel file where every person is listed on the left with the paper it should present on the right. And you are welcome to shuffle this list if it doesn't match your dates or your preferences of papers. But just don't get me involved in that. I don't want to be involved. So just, if you don't want the date or the paper, find someone and swap. It's better if you let me know, but I don't mind so much.
Each one is going to present alone?
Yes. So the way we're going to do it, it's a challenging thing, and it will mean that you will have to work quite hard on that, because we're going to devote the first hour for 3 presentations. So, I mean, it will take, we will change the break a little bit, but we'll have 3 sessions in a row of 20 minutes. Every person will present something like 15 minutes, and then we'll have 5 minutes for questions. And we'll just do it one after the other, and you'll have to be very prepared, because I'm going to stop you on the dot. And it means that you're not going to present everything in the paper. You have to work and figure out what is the most important part of the paper and present that. So I think it's a good practice, because if I give you like 2 hours to present a paper, of course you can do that, but to do it in 20 minutes, it's a bit more challenging. And then one of these 3 papers we will use for the second hour to go, figure by figure, in depth, understanding the concepts, understanding the methods, and doing it like a roundtable type of thing. Okay, so I will tell you in advance which one of these papers is going to be the paper that we're going to discuss for the next 4 sessions. Yes.
Not too much time to prepare for next week.
No, I think for one paper it's okay to prepare. The person that present, the people that are presenting the papers are not, I'm not going to challenge them on the discussion. I think it should be okay.
How much time do we have? When are we starting?
We're starting next Tuesday.
It shows fine with
It's
their
not
presentation.
enough time
Because
for
we also have other things
Yeah.
to do.
I
This
know.
is the only thing we are
Clear cycle.
Look, I know it's not ideal, but first of all,
6 pages of computational.
no. The first paper is special one, and the one who's going to talk to is only it's only the beginning of it. We're not going to go through the whole paper.
And also when you prepare for this paper,
Yes.
so you
The first paper is special. I will tell you about it. If you want, I can tell you about it now. I just don't remember the number, but I marked myself which page. I will send you an email until which page you need to read it. It's actually only the first few pages. It's not that difficult. I mean, it's a difficult paper, but it's not that. It's not the whole thing. The problem we have is the following: the schedule is already kind of been changed, and I cannot, because of other people that are presenting in the course, I cannot shuffle it anymore. It's kind of, I cannot now move Alex Binstock anymore. So we have kind of hard deadlines. Let's talk about it after the class, okay, about the details, because I want to also give you some real stuff today, or in the break. Okay. So last week we talked about all kinds of aspects of noise and coding. And I just want to finish this by presenting you few examples where this comes with some surprising results. So the first example I want to tell you about is the example of motor control. Some of you may have seen it either in the undergrad studies or last year in physiology. And this is the example of population coding, where you have a monkey who needs to move the hand to one of 8 targets. And what the people are doing are recording from motor cortex, from the motor cortex of the monkey responsible for the hand of the monkey. And what you see here is raster plots. Basically, like every dot here is a spike. And these are different repetitions of the same movement when the monkey moves the hand towards this location. Okay, so the monkey gets a go signal, and then he moves the hand with the handle from this location to that location. And so, of course, it's doing it with random trials, but then you organize the trials such that all the trials that happen when the monkey moves the hand toward that location are represented here. And what you can see here is 2 important phenomena. What's the first? What is the first thing that comes up when you look at these raster plots? Someone who hasn't seen it before.
I think everyone saw it.
Everybody saw it. Okay, so what you see is that you have tuning, right? So the monkey, when the monkey moves the hand towards one direction, you have more spikes than when it goes to the opposite direction. But the other thing that you see is what, in our context, come on, we'll never finish this way.
not exactly in accordance with
Right, the spikes do not repeat themselves exactly in a similar way, right? Every time the monkey moves the hand, you get a different spike train. And there are different reasons for that we can think of, but what I wanted to do is actually show you an example that we will have as one of the presentations, but I just want to give you one highlight, which comes from from the systems of bird songs. So when you record from the brain of birds when they are singing, there is a specific location in the bird's brain, which is called HVC, and you'll hear more details about it in the paper presentation. But when you record from a specific type of neurons that are located here in the bird brain, you see this very special example where you have the birds. What you see on the top is a bird, the song of the bird in a spectrogram way. So you take the waveform of the sound, and you make a spectrogram. So you look at the frequencies as a function of time, and you see that there are different. So this is what we call a syllable. Okay, this is the distinct sound that the bird is making during the song. And the song, when the bird sings, it repeats this thing several times, sometimes 2, sometimes 3 times. And what you see here is recording from different neurons, just as we did it with the monkey. So now the bird is repeating the song, and we are recording from a neuron, and what you see here in green is this neuron spiking during the song. And this is almost the complete opposite of what we saw from the monkey, right? So if you look at that, this is 10 milliseconds, and during this 10 milliseconds, this bird is spiking 4 times. This neuron is spiking 4 times, exactly at the same location with a millisecond precision. Okay, and now this is not an auditory. You could think this is an auditory neuron. So this is a neuron that basically hears the bird singing, so it's responding to the sound very accurately. But this is not an auditory neuron. This is a premotor neuron. This is a neuron that spikes before the syllable happens. It actually triggers the syllable. We can actually kind of show that in all kinds of different ways, but this is not an auditory response. So the neuron, or the brain in that sense, the bird's brain is generating a very precise output without having, anything, a clock or anything that will generate it. Okay, it doesn't always happen. You see, there are neurons that are more sloppy, and moreover, there are other neurons, like interneurons in the same area, that behave a little bit more like the motor neurons of the monkey. So they have some correlation with the song. You see that there is a pattern here, but they are not very, very precise. So this is, I think, maybe one of the best examples where we can show a neural system that can generate a very precise output spikes. And just kind of in a different context, I can tell you that the model that people believe happens here is very similar to the thin fire chain that we talked about before. Okay? Interestingly, in the same system, I don't know if it will work. Let me see. In the same system, there is this interesting behavior where you can actually show noise that is kind of planned. So we talked a lot about what are the sources of the noise. Is it coming from like the single channels? Is it coming from synaptic systems? Is it chaos? So here is an example where the noise exists, and I'll show you that not only it exists, apparently it has also a function. So what you see here is 2 cases where you see a bird in a cage, and in this case, you have the bird on its own. So the bird is alone in the cage. It's a male bird, and it's a juvenile male bird. Okay, and For some reason, the sound doesn't work here. Maybe I'll try to do it. Can you hear it? Okay, so this is the case, the first case. Now listen to the, try to listen to the second one. Who can say what's the difference between the two cases? Did you hear a difference? What's the difference?
You have to show me that.
No, of course, but no. Can you describe the difference?
The second case is faster and more female.
Okay. Anyone else? Okay, let's try again. It's a little subtle, but do you hear that there is some structure? Okay, so pay attention to the structure in the first case and in the second case. Okay. What's the difference in the structure?
The end of each syllable is
In the system, he's adding the small church in the middle.
Right, but so look at the beginning here. It repeats the same song twice. Okay, so that's the song. You see, it's exactly the same thing, right? So there are a little bit kind of noises in between, but when it repeats it exactly the same. Here, whoops. There is something similar, right? You can think that it's similar, but it's not exactly the same, right? So basically, the idea here is that when this juvenile male is in the cage, it's practicing the song. At a certain age, what happens is that it already knows the song, but it will kind of make all kinds of variation on the song when it's alone. But when it is with a female, okay, when it sees a female, then it repeats the song exactly the same way every time. So, except of all these kind of strange things in between. But when it sings the song, it sings exactly the same song. So you can see it here. We call it direct and indirect song. So an adult male, it doesn't matter when it sings, if there is a female or not, it repeats exactly the same song. So you see, these are 4 repetitions alone. These are 4 repetitions with a female. All of them are almost identical. However, if you get the juvenile one, you see that when it repeats a song, on its own, the length is different. Different syllables would sound differently, and it can kind of introduce all kinds of variation in the song itself. However, when you take exactly the same one and you record it with the female, it repeats exactly the same song to the female. So it already knows the song. It can control the preciseness of the song. but it chooses when it's alone not to do that, and when the female is present, it's doing it. Okay, so this is a really interesting case because there's another place in the bird's brain where you can record the activity of neurons, and you see the difference now. They flipped the order. So this is a directed one. This is with the female. This is without the female. And you see basically the differences in the precision in the two cases. So there's a lot of more noise here than here. And the kind of the model belief is that that this is kind of an area that generates noise during learning in order to actually change the song, so the male can actually practice and find, you know, different variation until it finds the song that it wants to eventually have. So there are cases in the brain where we think that noise can be even beneficial and actually being generated in order to perturb the system, preventing it from repeating exactly the same thing every time. Okay. Okay, so the last bit, I think we'll do it maybe after the we'll go into these details at the end of the somatosensation part. But when I asked you in the beginning, how can we actually probe this question in terms of like how can we actually tell when neurons have are using noise or not using noise, if the noise is a feature or a bug, this variability in spike train. Some of the ideas were like what I showed you now. Explore different systems where you present this, you present the stimulus again and again and record the spikes, and try to correlate the noise that you record with what happens in the stimulus. And we talked also about some ways to estimate the variation using information theory or things like that. However, as you know, we have better ways now to manipulate neurons with optogenetics, for example. So one of the directions that is happening right now is the following idea. So this is kind of just a sketch of the idea, but imagine that you have a mouse that is doing some task. And we'll talk about these tasks in the paper sessions more, and also in the end of the next lecture. So imagine that you have a mouse that is performing some task, and now you can kind of record the activity of neurons in the brain of this mouse while it's performing the task, just like we did with a monkey. Okay? And now imagine that you have some way, say optogenetics, to label some of these neurons that you think are relevant for the performance in the task. So imagine that here you can identify some neurons that these neurons, the activity of them correlate with what the mouse is doing. So if it says, yes or no, or with some measure of the performance of the mouse on the task, and you can transfect these neurons to express channel rhodopsin or halorhodopsin or something that lets you actually control them with light. Then you can actually go back now to the task and perturb these neurons during the task. So for example, you can look for neurons that are activated in some ways during the performance, during the task, and say, now if this neuron was firing, 20 spikes per second during the critical point of the task, you can say, now I will take these 20 spikes, but I will shuffle them in time when the neuron is repeating when the mouse is repeating the task. And then I can actually say whether what matters is that the neuron is spiking these 20 spikes, or whether the precise timing of these spikes matters or not. Okay? So this is where these things are going, and we'll see some preliminary examples of this at the end of the next talk, I think. So, yes, okay. So let's go now and switch to somatosensation. Any more questions about this topic before we go to somatosensation? Yeah, all kind of completely overwhelmed by the paper sessions and you can't think of anything else. Huh? That's my feeling. Okay. Anyway, so somatosensation. We'll do it pretty quickly, and then if you have, I mean, these are filmed also from last year. So if you want, you can look at more details on the lectures from last year, which are kind of slower in some ways. And you already have kind of experience with several sensory systems that you've seen before. So I want to mainly highlight the differences that the somatosensory system has from, auditory and visual and other systems, but also I want to use it because mice are highly, rodents in general, animals that are using the somatosensory system more than visual. It is now in the focus of a lot of research. So it's good to know some things about it, and I'm going to use it in order to come back to some of the questions that we just talked about right now. So somatosensation is basically a big name for different systems. So you have a system that is responsible for touch, for pain sensation, for temperature, and for proprioception, which I don't know how much you talked about it in the motor system, did you? Okay, so this is like you have a sense of where your body is in the space, like what is the angle of your joints, what is the length of your muscles. So this is also part of the somatosensory system. And we are going to focus mostly on touch, and then later on pain, where Alex will tell you about it more. It's a very important system because it is important for you to know the integrity of the body. If you have if you have a cut or an injury, where you are in space. And apparently it's also kind of very important in terms of pleasure. Maybe I'll touch on that a little bit. I think Alex will talk about it as well. One of the things that is very special about it is it's multimodal. So because it's not one type of stimulus that is generating the sensation, it is somewhat different from visual, which is the same type of energy that drives everything. The other thing is that I think you would appreciate it, that in some ways, we don't pay a lot of attention to this sense of touch so much. Like we tend to think that we are visual people, and we are very auditory because we're using language through speech, and touch is something that is less important, but it is that when you go to a museum and it's a highly visual place, you always want to touch things, and unless you touch them, you don't really know that they are there. You kind of the sense of reality is almost always through touch. It's very easy to do virtual reality using vision, using auditory. It's extremely difficult to do it through touch. Although I'll show you some examples where it's like, these examples where you can fool this system. And at some point, because of that, many people are relating that to the notion of self. You know, who am I? How do I know that this is my hand, and I'm controlling this hand, and no one else is moving my hand? And you know that some of the problems that you have through psychiatric disease are when you think that I hope you don't, but people who have that sometimes think that they are not controlling their body. So this is extremely important to the notion of who am I, what is me, and what is others, and it became also a very useful tool, this system, in order to access these questions. Okay, and so the classical example is this rubber glove hand experience, which we probably all saw, but just for the sake of the people who haven't, it's always fun to watch. You can create this illusion where you make people feel that this rubber glove is part of their body by generating this touch and visual sensation at the same time, right? So this is kind of something that is always fun to watch, but it is actually now extremely used in cognitive psychology, in systems of virtual reality, where you basically create avatars. People kind of wear these glasses. They see themselves from behind, and for example, you can touch their back while you touch the avatar back, and you can kind of control for where they think they are in this virtual reality, and kind of do all kinds of manipulation through this system to explore this notion of self. Who am I and who is someone else? Apparently it's pretty easy like that to create this illusion that you are somewhere else, that you're kind of out of your body, or that some other things are part of your body, just by this coincidence of touch and vision, which can bring us back to all these issues with dendritic spikes and stuff later on. Okay, so like any other system, the classical view was that you have like a feed-forward path of information from transduction. So you take the physical energy, which is in our case, say, pressure or vibration, and transduce it into neural signal, and then you transmit it into the brain. You integrate it on the way, and you create perception. As you've seen probably in other systems, in the visual system, the modern view is also taking into account the fact that you have a lot of feedback. So you have loops, that information is not flowing only in one direction, but it is a lot influenced by the other direction as well. And we'll see some example. One of the examples that I'll show you later is the example of this vibrissa system of the rodent. So yeah, they have these whiskers, and they use a lot of these whiskers to learn about the world. And you would think that the input comes from the vibrissa, feed forward to the cortex. But as you can see from this diagram, almost at every level there are, loops that are closing. There are loops of other directions that the information flows. It can actually go very, very quickly through subcortical areas back to the muscles that move the whiskers. So the information doesn't have to go all the way to the cortex in order to for the rat to react to things that happen. Okay, the loops are closing very, very quickly, and this, of course, generate a very complicated system to study, because you never know, you know, how the information arrived to the cortex. Did it arrive this way, arrive this way, and you know, what was the relationship between these loops that happened so quickly here on the information that eventually arrived to higher areas. So it makes things very complicated. So I want to. Okay, let's stop here before we continue. So how many of you really can describe to me what happens when I, you know, say I do that? I kind of touch the tabletop and I tell whether this is smooth surface or rough surface. Who can tell me that very, very quickly? Like not in many details, but
Different type of photoreceptors which are different, like rapidly adapting, slowly adapting, and the combination of them will generate the receptor.
okay. How many of you knows what's been explained now? Okay. How many don't know anything? Okay, so we'll do some mixture of the stuff. So just so some of you will find it kind of boring now, but anyway. So basically, the system is working in the following way, which is not so obvious to begin with. So we have on the skin, everywhere on the skin, we have some sensors, just like we have in every other sensory system. But the difference is that in visual system, you have the retina. This is the organ where the receptors are, where light is transmitted and transduced into neural signal. But in the somatosensory system, basically the skin everywhere on your body has these sensors. So it's much more, it is not one organ unless you consider the skin as one organ. So you have everywhere these sensors, and they transmit the information to the spinal cord. Okay? It flows in the spinal cord. We'll see the details how it flows in the spinal cord. At some point it goes to the brainstem, then to the thalamus, and then to the cortex. Okay? And we're going now to go through this path very, very in much more details. Okay, this is what we're going to do now. So first we're going to talk about the sensors, and we have different sensors for different type of energy. So we have for touch, as we said, for physical pressure. We have for temperature, different type of receptors. We have different type of receptors for pain, and we have different type of receptors for proprioception, for like the length of the muscle, and for the Golgi, for the angles of the joints. But we're going to focus on those, and we have. So if we take some example here, well, I'll show you later these receptors. But the idea is that in the skin you have a mechanoreceptor. Okay, and then whatever you do in terms of touch to the skin, this will activate the receptor. The receptor itself is connected. It's either really just the end of the nerve of the axon that comes from the spinal cord, or is a neuron, or is a cell that is connected to the axon, and it generates an action potential in the axon. Okay, so the touch will generate an action potential by opening some ion channels, and this action potential will flow all the way to the spinal cord. Okay, so these are these type of neurons that kind of don't have dendrites, or dendrites are not really important. Everything happens in the axon. The neuron is just sitting there, providing kind of proteins and stuff, but it doesn't actually participate. The body doesn't participate so much in the preparation, in the integration of input. So how do we actually how do we generate this action potential? So there are different ways. One is just mechanical force of the membrane can actually change the structure of the protein and allow ions to flow in and generate action potential. But there are all kinds of other ideas. For example, you can think that there is something that is connected, some tissue that is connected to the protein and open and close it, similar to what we have in the ear. And the truth is, or you can think that it's going through some, second messenger. So you have some sensor in the membrane that will activate the protein from within. The truth of the matter is that we really don't know much about it. For some mysterious reason, I never figured out why. But the first paper that really identified these proteins was published in 2010, and it is called. It's a paper that we are going to show in one of the presentation. It's called Piezo 1 and Piezo 2. These are the names of the channel. Who knows why? Yeah, so what piezoelectric devices do you know? So there are basically ceramic types of materials that when you apply voltage to them, they can actually change physically. They move. Okay, it's just like small motors if you want. And in the other direction, it also works. So if you move them, they will generate voltage. So it used to be very useful to put on like guitars, because then the vibration of the strings would actually transduce into electrical vibration that you can actually amplify. So they call the protein Piezo 1 and Piezo 2. And these are the first two ones that we know of that transduce physical force into voltage. We don't know yet how they work. I mean, we just know which one are the proteins, and we can show that by expressing them in cells, they react to pressure, but we don't know how they work completely in terms of like the molecular changes. I already told you that there are different type of receptors. One of the very interesting version of them are called the TRP channels. Do you know about them? The TRP channels, it's a very interesting story. I'm not going to tell you much about it, but the first time they were actually discovered are by Baruch Minke, who is actually a member of ELSEC, and he found them in the fly visual system. And it turned out that it's a huge family of proteins which are extremely important for many, many aspects of sensation, for thermal sensation, for pain sensation. And Alex will tell you a little bit about them. But the TRP channels are extremely important in those aspects. And those ones are understood a little bit better than these piezo ones. Okay. So let's just mention which receptors do we have. So we have 4 types of touch receptors. 2 of them, and we have different ways to classify them, these 4 ones. But in short, we have 2 types. 2 types that are very, very close to the skin surface. They're called Meissner corpuscle. Corpuscle בעברית זה גופיף. They mean something like, you see it here. Meissner corpuscle and Merkel discs. And you see that the skin is kind of has these grooves. So the Meissner corpuscles are sitting just under these grooves, and the Merkel discs are under the heels, if you want. So these are the 2 types of the surface receptors. And deep in the tissues are 2 other types of receptors. One of them is called Pacinian corpuscle. It's another corpuscle. It looks like an onion. and you see that it just kind of sitting around the nerve ending, just around it. And the other one is called Ruffini ending. It's another type of ending of the nerve over this structure. Okay? And you see that near to every one of them there is another label. This is RA1 and SA1, and this is RA2 and SA2. And we'll talk a little bit about these in a second, maybe after the break. Okay? So let's take a break and then we'll talk about it. כן. קיבלת את התף כשהמאמר ראשון, אני מבין.
גם, סתם אני אצטרף.
כאילו זה לא משנה.
זה קצת מה... לא, זה סתם זה מאמר, סתם קראתי את זה. אני יודעת אם זה יעזור. זה גם טוב יהיה כשלך. אני יודעת אם זה
אתה עשיתם כאילו לפי הסדר של אני אולי צריך ליישר. מה? להקים זה. כן, עד איזה עמוד? הנה רגע, אני בוא.
כאילו, אם זה הדבר היחיד, זה לא הבעיה. מה? כאילו, יש לי עוד קבוצה של לעשות.
למה לא תבוא?
לא יודעת כמה, יכול להיות שאני רוצה לבדוק כאילו כמה, לא יודעת כמה אנשים, כאילו אני קודם רוצה לדעת מה ההיקף. לא יודעת כמה אנשים ישתו
לא, אני לא צריכים את שישה, אני אמרתי לכם, בבאה אלה אנחנו צריכים אחד לכל...
שלושה, לא?
מה?
שלושה, לא ארבעה, לא?
אנחנו עושים שלושה מאמרים בכל סשן.
ויש שני סשנים שלא,
שניים, אבל אתם לא צריכים לקרוא את כל השישה מאמרים. מי שמציג צריך להציג את המאמר שלו. וכל השאר צריכים לקרוא שני מאמרים מתוך השישה.
אז אני, כאילו, אם אני מציגה, אני לא צריכה לקרוא את כל השאר מאמרים? כאילו, בעיקרון אני מעדיפה לקרוא, אבל אני מתכוונת מבחינת
אני לא כאילו...
את השאר הולכים להזכיר?
עכשיו אנחנו עושים כזה דיון פתוח, אז אני לא...
אני מעדיפה לקרוא
מבחינתי זה בשבילכם, זה לא בשבילי.
זה העניין של העומס.
אני רק רוצה לראות... מה
זה כזה כל הפיתוח של...
כן, של
כן, זה היחיד שכל באמת בעניין
כן. הסגנון מאוד למרות שהיה במחשב החדש שלך מאוד שיין
כאילו זה גם חלק מהעניין של
1489, זה מה שכתוב.
אני עד לדריביישן?
כן.
אוקיי.
מה?
איך נשארת פה?
כאילו זה עניין שאני כן רוצה לקרוא את זה, פשוט כאילו יש עוד מלא דברים ואני רוצה לעשות את כמו שצריך ולא לעשות את זה
אני מבין, אני מבין את המגבלות ואני לא, זה לא בחינה, זה בשבילכם. אמרתי לך, מבחינתי, זה לא קורס...
ויש לכל אחד רבע שעה
זה לא טעים.
זה משנה את ה-26 עמודים.
אני יודע,
ראיתי את זה פשוט לקחו אליי תודה.
מה זה לא טוב לך שנה? אני קיבלתי אותה, זה לא אני הכנתי. זה הרגע לא שווה. לא, זה לא שווה. אז מי בחר לי בכלל הפרוגלוטן? אני עושה עליי
ואז אוכל דברים שליליים, אז זה אי שלילי.
זה גבינות
זה
קשות,
בסדר.
It's the same first thing.
זה לא עושה כלום.
אז לאט לאט תורידי הכל.
אז לא נגיד תמיד את זה.
בסוף אני אוכל כזה מים אוויר. שמעת פסה. כזה רק תראי שכבר נפלו מהם.
יש דרך, אני מבין מה אתה שנייה, יש דרך, לא צריך להגיע לאינטגרלי של זה.
לא מנסה להגיד את זה יותר ברור.
איזה מגניבים תהיי פוטוסינתטית?
זה לא קשור לאינטגרל, זה גם
אני מתחילה מחר כי אנחנו נוציא אותו אני מתחילה מחר
חברת אקסל, המאמר שנמצא באותה שורה שלי, זה המאמר שבינתיים אני עושה נכון, כל עוד לא מחליפים אותו. אני מתחיל לעבוד.
הוא באמת מפגר,
מה האינטגרל?
אני זוכר, זה המאמר שנראה לי סבבה, רק לא יכולתי להיכנס אליו ולראות אם הוא אלקטרופיזיולוגי או לא. זה על סומטו סנסור.
אני עובדת פה משתיים, כי ההגדרה שלה הייתה העבירה שאני לא הולכת לבוא לך, אבל יש לי שעה אחרי
כן, אבל למה יש ההגדרה של הבנתי, בסדר, נראה לי למעלה.
יש את ה...
לא, זה בסדר גמור.
בעצם, אבל כרגע גם סדר גודל יעזור, זה כולם כאילו בשבועות הקרובים או...
רגע, אתה...
לא! יש דבר שאני חושבת
זה נספר לך כאילו לשם.
המאמרים כולם יצבו בשבועיים הקרובים?
לא, וואו, זה 4
זה 4
זה לא, זה 4 סשן.
4
סשן, כמו שאמרתי.
12. כמה אנחנו?
24. פשוט המון.
עם כל הדוגמאות, זה לא אבל על הדוגמאות. אני עוד לא מספיק מתורגל. זה לא מה ששאלת, הכניסות הזה כמו שישי הזמני. אבל לא מתחילים להיות מתורגל. יומי ושישי.
What? Do you want to sit next to us?
כן, אז באמת, אז אוקיי, אז יש לכם עכשיו, אנחנו חיים באריאל. המשלים האורתוגונלי לוי
Yeah, I don't really.
כל המסה שלה היא בקבוצת מרחב הזה, אז עם פרופר באיזשהו זמן, כי כאילו...
Do you know who have a group, who don't have a group? I mean, do you know who already have a group and...
I think that everybody already has a group except, I mean, 2 people told me that they don't have, which is Way and Safa, and I told them to talk to each other, and then you wrote to me. So it's kind of strange, but...
So there's maybe another one.
There should be another one that doesn't have a match. Maybe I'll ask just to my staff. But I don't know if everybody is Okay, I'll write to them. I'll write to them.
If you know when it
So,
if you know what is my date.
you didn't know that?
I think you just know.
שלי לא. שלי אפילו בכלל לא סיפרתי עליו כבר מהקוצים.
כן! יואו!
You don't
It's like
have
I
to.
told her, I was every night tell someone like I want to because you're talking about me. It's just easy one. Like
You don't
it's broken and you never found it.
Okay.
אז תגיד, שלחתי תאים. זה לא, אין פה מה להבין תאים, פעם זה אוכל
Close to your home, like, just to eat.
לקו האורך.
This is for me, you didn't tell me.
לא, כאילו לקו האורך פעם אחת, ואז יש לך כאילו שתי סירות כאלה, נכון? ואז אתה לוקח כפית, ואתה מרוקן לו את האלו. אז גם זה לא מתאים. ואז הם יכולים את הכאילו, זה נראה... נעבור את החוץ. זה לא מדהים לא, את פשוט לוקחת אוכל, זה כאילו זה מים, זה טיפה טעים. לא, כל המנצפות זה מים.
החלקים של הירקות שאתה פחות אוהב לאכול, אתה משאיר אותם לתוך כזה, אם אתה פשוט חותך אותם ותשאיר אותם לאוכל שלך. כאילו, לא יודע, שזה את העלים החיצוניים, את השישה העלים החיצוניים של החתיכה או של הסלום.
אני עושה את זה ממש, אני ממש מציעה פלפל, אבל הגעתי עוד לתת
No, by the way, it's
No, that's like...
אז אני ממש מטבעת מטורף. אני מבינה, אני הרבה יותר אוכלת סלט.
No, this one.
אני הרבה יותר נהנית מהסלט שלי, אם אין לו את כל החלק הזה. No, it's not this one.
Oh man, no, it's just...
אבל תקשיב, לא סיימתי. סויה, קצת גילן, וטיפה למים ולגדר כזה, וגם פירות חמים. אני מבינה שהיא קיימה אותי. למה אתה לא מסכים לזה שזה סלט? זה ירקות. אבל זה ירקות חצופים בתוך קערה, זה סלט.
עם חמת נופלים ודברים אחרים.
חמת נופלים זה כמו להוציא טחינה לסלט, רק בגרסה משודרגת.
זה ממש טעים, אבל אתה יכול לתמיד להגיד עוף פירות. נכון, אז אם אתה חותך גם עוף ומכניס את זה פנימה, הגרסה היא פשוט עושה מוקפד, אתה זורק את זה סלט. לא נכון, די! זה לא מתאים. אז הרבה כמו
וואי,
אתה חותך אותו
שוב הגענו לזה.
לקו האורך, אתה לוקח כפית ואתה מרוקן את כל הסוגינס שלו. ואז אתה חותך את זה, אתה שומע את הקליפה. קטנים כאלה שזה...
אז למה פתאום מקלפת את המקום?
את חשבת על זה?
למה מקלפת על המקום וזהו? של החוק.
אני לא רוצה קליפות על המקום.
זה מה שקרה בפועל, אתה כאילו הולכת לפעמים אתה תתחיל לרדח, כאילו רק את זה. בסך הכל כל
התחנה שבסופו זה לא בטעים. למה זה לא טעים? עם כל הבלגן, כאילו, אפשר לא לטעים.
כאילו, אני לא רוצה לא לאכול אותו, אני פשוט תיקול.
אני לא אוהבת את המלפפון.
זה מה תגמריות.
אני אוהבת את המלפפון, אבל למה אתה לא
לא, את לא אוהבת 65%
זה לא מלפפון, זה רטוב כזה. זה
זה כן הקובץ הנכון, זה פשוט...
את הקובץ הנכון?
כן, זה הרצאות היסטוריות. לא, סתם זרקתי.
אני לא פתחתי את זה, אבל אלכס כבר פתח את זה עכשיו וראינו שזה כאילו...
יש לו שני גיליונות.
זה מה זה שיר?
זה ממחר.
לא, עכשיו אני צריך לעלות אותו עוד פעם.
No, this is the one I thought about, I'm sure.
אני לא יכולה כאילו להגיד להם, סליחה, החלטתי
Okay, so just before we continue, two things. I uploaded again this Excel file. Now it has two pages. One of them is with the papers, just as before, and one with the date of the lecture, so you can actually match where do you have to talk. So again, so the Excel file now also have the dates of the lecture, so you know which paper session comes at which date, so you can actually match when your paper is coming. It's modulo 3, so you can just do that. And I will send you the...
Wait, there are 3 lessons until we will finish them?
Well, no, so we have, I'll show you, okay, here it is. So we have, we are here today, somatosensation, and then you can see. Let me do, okay, so we are here today, somatosensation. Thursday will be also somatosensation, and then we have 4, like the next 2 weeks will be paper sessions. And then we're doing pain, and then there's another paper session on pain later on, and so on and so forth until we finish.
Do you have it written somewhere what are papers 1 and 2?
So the papers are just going like 1 to 3 is paper 1, 4 to 6 is like paper session 2, and so on and so on. Okay. And the papers are here, so it's the same Excel file. And now, one more question. Although you have 24 students, it seems that Alex doesn't have a partner for the assignment 1. So is there anyone else that doesn't have a partner? It's kind of strange. Okay, Alex, I'll let you know. I'll write to everybody and I'll let you know. Okay.
Can we do it in a 3?
I'd prefer not, but if there won't be a choice, then I'll pair him to another pair. Okay? Okay. So we talked about the Meissner corpuscle. So these are just staining to show you how they actually look like. And this is these are the Merkel discs. So you see it's like small cells that's sitting on top of the axon terminal. And when you apply pressure, these cells actually cause the terminal of the nerve to spike, and the spike goes back in the axon towards the spinal cord. So it's the opposite of motor axons that are propagating from the spinal cords to the periphery. These ones are carrying the spike in the other direction, in the reverse direction. So okay, this is less important. Wait, so I just want to show you one more thing, which is important here. You see that, for example, you saw it also in this picture. Maybe this is nicer to show, sorry. So this is an axon that goes to the spinal cord, and you see that it branches here. It has branching, right? So it's kind of, it occupies some space, and you have different Merkel discs over here. Okay? What does it actually mean in terms of our sensation? What's the interpretation of that? Sorry? Low. I don't know what's low and high, but it basically means that if you sit here and listen to the spike, you cannot tell if the spike arrived from this Merkel disk or from this Merkel disk. Okay? Once the axon is spreading, okay, and it occupies a space, every touch at every one of these places will generate a spike and will kind of propagate. And once the spike is in the axon, there's no way to tell which way it came from. In what sense? Yeah, I mean, these axons that goes in this direction, they don't have terminals that release neurotransmitter. And the axons that are going from the motor systems, they are synapses on the muscle, and they are making synapses. They are very, very similar. Yes, very similar. There are different type of those, and I'll show you, but they are very similar. Yes. Okay, so in terms of resolution, you can see, so in similar to what you have in other sensory systems, you can define a receptive field. Okay, what would be a receptive field? It would be like in visual system, a receptive field is the area in the visual field that activates a neuron. So here it is basically the area on the skin that activates the axon or the neuron, if you record from the neuron, downstream. So in this case, you see that the receptive fields of the superficial sensors, the Merkel and the Meissner corpuscle, are pretty small at least on the hand. They are not very big, so they are very small. And if you zoom into one of them, you can see hotspots, and the hotspots are the physical location of the sensors. So if I touch, with a needle on this spot, I would be more sensitive here than here, because basically what will happen here is that the skin will stretch a little bit and activate this Merkel disc. But if I'm on top of it, I can apply the force, it will be more sensitive. But these are kind of nuances, and the deeper layer sensors, the Ruffini and the Pacinian corpuscle, have much larger receptive fields. So for example, you see this Pacinian corpuscle, its receptive field, it's basically half of the hand. Okay, or this one here. So they are deeper in the skin, and there are less of them, and basically this makes the receptive field bigger in terms of area. I'm going to skip that and that, and then you can actually record the size of the receptive field if you want, or the resolution on different locations in the body, and you see that the best resolution that you get is in the fingertips, in humans at least, okay? But it's kind of surprising. So this is like in millimeters. So the idea is that if you ask the person to tell, so you take like two needles together and you place them on the skin and you ask them to tell whether this is 1 or 2, and then you change the distance between them and you measure the difference. Basically, if it's a static touch with a difference, the resolution is pretty poor. It's on the order of a millimeter, 2 millimeters, or maybe 3 in the fingers, and it's like in the back it goes to 5 centimeters. So you touch two locations with 5 centimeter distance, and person cannot tell if it's the same location or not. Okay, so the resolution is very poor, and this doesn't make a lot of sense because we're doing, we can do very, very fine things, and partially the explanation to that is that you have other ways to tell the difference, which has to do more with frequencies. I'll show you in a second. I'm going to skip that. This is interesting but less interesting for us now. So partially, the reason that we can do better is because of the temporal aspect of the encoding of these receptors. So you remember that I classified them as SA1 and RA1, SA2 and RA2, and this stands for rapidly adaptive or slowly adaptive. So what you see here is a case where we record from an axon. We record from an axon that is connected to Merkel disc, and this is called the slowly adaptive receptors. And you see that if I apply a pressure and hold the pressure for a long time, the receptor, the axon, actually spikes all the time that the pressure is located there. So long that I apply the pressure, the axon continue to spike. Okay, more or less at a regular frequency. If I increase the pressure, then at the beginning I have some high frequency, but then the frequency becomes more or less constant, and it continues so long that the stimulus continues, and even with a strong stimulus, this happens. Okay, so basically there is a change in the spike rate when the stimulus starts, and it adapts, but it adapts slowly. The firing rate adapts very, very slowly. So the time scale here, I forgot what it is, but it's kind of terms of seconds. Okay, so this is like slowly adapting. And this is in contrast to the rapidly adapting ones, for example, the Meissner corpuscle, where you apply a pressure, you see that there is spikes on the onset and then spike on the offset. So when the pressure is released, there's also a spike, but during that time there are no spikes. What are these? So these are spikes, right, just like these ones. And there are also these one here. What are these? Spikelets? On axons, usually we don't have spikelets. No, it's either there's a spike or no. There's not like spikes on different heights on the same axon. What else can it be? Right, so this is apparently you put the electrode next to an axon and you measure these spikes, but you also pick up on the same electrode spikes from another axon, which is apparently connected to a slowly adapting receptor, and then it kind of spikes slowly then. Okay, so you see that if you give a ramp, you change the pressure with time. You have so long that the pressure is changing, you have spikes. Once it becomes constant, you don't have spikes. Okay? What does it look like? Mathematically, how would you call it? Right, it's kind of a derivative of the pressure, if you want. Okay, when you change the pressure, you get a response. So this.
Excuse me, could you explain the number on the left side? Maybe I missed that.
The number here?
Yeah.
I don't think I said it. I think it's like how much do you move the thing that you kind of pressure with. So if you only move it by 270 microns, you touch with the needle and then you press. Okay? If you move it like 0.3 of a millimeter, you get response, and if you move it by 1.5 millimeters, you get a lot of. This is basically corresponding to that.
Yeah. So basically, it represents how much pressure.
Yes.
And on the right, it's the speed.
Yes. Okay, so the first person to, well, I don't know if I'm not sure it actually was the first one to, the first one to do it with a touch is Vernon Mountcastle. A very famous person who basically today people believe deserved the Nobel Prize, but he didn't get it. I'll explain to you later why. But these are basically what you saw before, are basically pictures from his oscilloscope while he was recording those. So you can see here the signal of what he was applying, and then the responses of the spike. You can see them here as well. So these were the first recording, and I'll get back to him later. This is a picture from the book of Kandel, trying to explain how you use these temporal responses in order to tell differences in tactile stimuli. So what they do here is they put a rotating drum with braille letters. So this is a sequence of the letters, and what you see here are responses from an axon of slowly adaptive, rapidly adaptive, slowly adaptive 2 and rapidly adaptive 2 sensors. And it's a little bit counterintuitive, but what you see here, for example, is that once the dot, is touching your finger, you get response, so long that the dot is there. Okay, and then when you have 2, you have this, that response, and in contrary, when you get these rapidly adaptive ones, you have like these spikes that are kind of, because the dot is kind of has some friction, and basically when it moves, it's kind of vibrate your skin. You get these kind of vibrations, okay? And in some ways, you would think that this would be better to encode the signal, but it actually turns out that these ones are better. And the ones that have like very big receptive fields, this Pacinian corpuscle, which are very sensitive also to vibration, so they're rapidly adaptive. They kind of signal this, this friction, but not so much what is the signal. So we tend to think that, for example, these ones are very good in terms of, like, if you want to tell the texture of a surface and you move your hand, you have very good signature of the special frequencies that there are in the texture, but not so much the fine details of the special resolution. Okay, so it's good for big surfaces, but not so much for very detailed, precise location. You can also see it from here. So what they do here is that they give, say, a sinusoidal wave. Okay? And if you have a rapidly adaptive receptor, it can actually follow the frequency of the wave, because the wave is changing and it reacts to the changes. But if you have a slowly adaptive one, it doesn't react very well to the changes. So what you see here, you see here two things. Here on the x-axis, you see the vibration of the frequency of the stimulus, and here you see the threshold. When you apply this threshold for a human, so you put like something that vibrates on the hand, and you tell the person to tell you when it feels the vibration. Okay, so in order for humans to tell the vibration at, say, this frequency, which is, 200 hertz, you see the threshold is actually quite low. You don't need a big amplitude. So you need something that vibrates very, very lightly. You can detect it immediately. In order to detect a vibration at a lower frequency, you need higher amplitude. So it needs to vibrate more in order for you to feel it. And so this is a perceptual threshold as a function of vibration. And here, they kind of do the same, but recording from axons of different receptors. So you see that what happens is that you have, each receptor has a different curve. So this is, for example, the curve for the Pacinian corpuscle, So at low frequency, it needs a very high threshold in order to activate this Pacinian corpuscle. So it's not very good at detecting low frequencies. But at high frequencies, it has very low threshold. So apparently this point here matches quite well with this point here. So if you want, you can think about it as like the spectrum that you have for different cones. Okay, this is like this is a cone that is sensitive to high frequency, and this is a sensor that is sensitive to intermediate and to a low frequency. Okay, and by combining these different receptors, we can kind of build a spectrum just like as we do with visual stimuli. Okay, but this has also a very interesting consequence that I don't know if I'll talk about it maybe in the next class, but I'll just highlight it now. This means that if you stimulate the skin with very low amplitude at a very high frequency, you know that the only receptor that is responding is the Pacinian corpuscle, right? So if you record any response, it has to come from these ones, because these ones are completely off in terms of the response properties. Okay, and this intuition allowed Mountcastle to actually probe things in the cortex. Basically, the mapping, which was very difficult from the hand to the cortex, you can actually do some of it by saying, now I'm stimulating only with this stimulus, and I know that everything that I'm recording has to come from these receptors, okay, at this location of the skin. Okay, and this actually had interesting consequences. We'll get to them back later on. Okay, what happened? Okay, so let's talk a little bit about the transmission. So once the spikes are in the axon, they have to go back to the spinal cord. And basically, the axons do not go all the way to the spinal cord alone. What they do is they bundle into what we call nerves. Okay, and this is like a cross-section of a nerve. And you see that in the nerves, the axons are kind of bundled up in bundles. And most of them are myelinated axons. And Aviv can tell you much more about that than I can. But this is a cross-section of a nerve like that, and you can see the individual axons wrapped up by myelin. And you see already here that there are different diameters. So you see there are big axons, and there are smaller ones, and there are very, very small ones. Okay, there are different sizes of these axons. So this is an illustration of those. I assume that you all know about this transmission through node of NVS. I don't want to get into that, although I think it's very interesting. This is again a cross-section of an axon that you see this wrapping of myelin around the axon. So the only thing I want to tell you about it is that you would think that node of Ranviers are just hotspots that don't have myelin and have sodium channels. This is probably what you learned. But it turns out that this is a very complicated structure. So for example, if you stain for sodium channels and potassium channels, you see that there is a very clear segregation between where the sodium channels are and the potassium channels are. And I remind you that this is 2 microns. So you would think this doesn't have any, well, why would you do that? I mean, in a 2 microns area, this is completely isopotential. There shouldn't be any difference. Why would you organize that in such a way? And it turns out that this makes a difference. It's not so obvious, and there are molecular machineries. So there are molecules inside the axon, inside the membrane, that make sure that the sodium channels are in one location and the potassium channels are in different locations. And when you perturb these molecular mechanisms, the transmission is disrupted. It's not so clean as it is with when this is actually properly done. So you see there are these molecules here that make sure which one is where. And for example, this molecule, this Casper2 molecule, where you introduce some mutation to it, you can cause autism. Okay, how? No one really understands. Okay, but it's kind of very, very strange. It happens. Okay.
How do you, I mean, did someone try to cause it?
So I think it, if I'm not mistaken, it happened in the other direction. So you do gene analysis and you find the mutation in autistic patients. Then you can introduce the mutation into mice, and then you can then it's like a gray area. What is an autistic mouse? But there are mouse model of autism, and they have some features that you can connect to autism. For example, some repetitive behavior, some social interaction that is not so not as typical as other mice strains and stuff like that. Okay, and this is not the only example where it happens. There are like, I think 12 that I know of. I don't remember all of them by heart, but if I'm not mistaken, specific mutations from humans that were done in mice. Okay.
So it has to do with the conduction?
So it's actually it's not clear. I don't know. I think that this specific molecule, as far as I know, the only function that is known about it is in this organization of the node of Ranvier, but it's not only the node of Ranvier, it's also the initial segment of the axon where the spike is generated. So it could be that the reason is there. Okay, so what happens when you apply? Say you kind of take one of the, you take an electrode and you put it in the nerve. It's a very wide electrode. It doesn't actually detect individual spikes, but you put it in the nerve and you stimulate the skin. So if the stimulus is very small, very gentle, that you almost don't feel it, okay? What happens is that at some delay after the spike, pretty short delay, you get this wave of activation in the nerve. So we call this wave A alpha, and it turns out that this wave is carried out by these large myelinated axons that are called the alpha. So the one in the sensory system they call the 1A axons, and they are myelinated, they are large, and they are fast conducting. So you get a short delay, and they are very sensitive. Okay. If you give a bigger stimulus, then you get another wave. A little bit afterwards, you see another wave of activation. And then yet, if you give another one, even higher, that it starts to feel like painful, you get another wave, this A delta. So there's A alpha, A beta, and A delta. And these are classified as different diameter and myelination of axons. And they are typically also connected to specific receptors, as we'll see in a second. And then if you make a much larger stimulus that is painful, really, really long time afterwards, you see this wave of activation, which is smaller. I think this is seconds. So I think this is something like 2 seconds, if I'm not mistaken. What?
What about A gamma?
I don't know. I think the gamma is actually saved for the gamma motor neurons. I don't know why they skipped the gamma. Okay, so these are what we call the C fibers. These are the fibers that are unmyelinated, and you know that if they are unmyelinated, they are much slower to conduct. They also have very small diameters, and these are usually the ones that are responsible for pain and for thermal sensation. So you know this fact that when you touch a very hot thing, you might kind of respond too late, so you already get you get hurt by the time you feel it, and partially the reason is because the conduction of these signals are relatively slow. So just to put some numbers, I already showed you these pictures. By this myelinated 1A neurons, they can conduct up to 120 meters per second. Okay, and the C fibers are around 1 meter per second with some variation, and you see the difference also, the order of magnitude difference in the diameter and the myelination. And what you have here is a correspondence between the type of the sensor receptor and the type of fiber. So the mesenchymal corpuscle are connected to the alpha and A beta, okay, and so on. So all the ones that we talked about, but you have other, for example, chemoreceptors that are responding through C fibers. So when you have pain because of, say, chili pepper that you touch, okay, this will be carried by these fibers, and it's pretty slow. Okay?
Do you think that's a good reason for why it's slow? I mean,
You mean something about evolution that I will tell you? I once made this mistake in this class and said that I don't believe in evolution. Not going to repeat it. I don't know. I mean, you can only kind of.
It seems like a disadvantage, not an advantage.
Well, I mean, disadvantage in terms of like that pain should be carried out very quickly.
yeah. So maybe there's a reason for this, maybe, or just because it's older, I don't know.
I can only speculate just as well as you speculate. I don't think I have any advantage in speculating on that on you. Really, kind of. I don't think that we have only any facts that can support any view of that. But probably it is true that it started, systems started unmyelinated and then became myelinated, and then why did this not kind of propagated also to be myelinated? This is just a speculation which I can't answer. Yes?
Sorry, why would pain from chili be carried through a mechanoreceptor?
No, chemoreceptor. Ah, sorry. No, that's true. Sorry. That's a mechanoreceptor. That's a painful mechanoreceptor. I meant, so I think the chili one would be probably through the heat, not receptor. And then the A delta, right, you're right. But the warm is, and the warm are the C. You're right. Sorry. Okay. So what happens in the spinal cord? You already talked about spinal cord in the motor system. So this is basically the reflection of the motor system, just in the opposite direction. So you know that you have different segments that are labeled by the vertebra along the spinal cord. One interesting thing about it, I think this is not what I wanted to show you. Yeah, so I kind of I skip that because you already know that from the motor system. But one interesting thing that happens is that these different sources of inputs. So, for example, input that comes from the muscles or input that comes from free nerve ending, they are getting through the dorsal root ganglion, okay, into the dorsal horn, and then there are different routes through which they propagate into the brain. Some of them, of course, are closing a loop. For example, for the 1A reflex, for the yeah, for the stretch reflex, for example, so you would get like an output from a muscle spindle that goes all the way through the dorsal horn, and then will synapse directly on a motor neuron that goes back to the same muscle. So you already know that from the motor system. But each one of them keeps their own track. Some of them will cross to the other side. Some of them will change in the same side. But what I wanted to show you is that we have this interesting mapping on the body, which we call dermatomes, because say all the inputs that are coming from this area of the skin are mapped through the T9 segments. Okay, so in a way we can kind of classify all this area as one area of the body that is mapped through this sequence, and these are called dermatomes, and they have this consequence. You know that there is this disease called Shalveket Hogeret, and this disease is carried by a type of a herpes virus that gets into the system through the ending of a nerve. So you have a cut, and then you get kind of infected by this virus, and this virus is traveling back on the axon. So it's picked up by the axon, and then it goes back, and it will actually go here and transfect all the neurons in the same ganglion. Okay, and then it kind of sits there, sleeping, whatever. And when the disease breaks, they generate a lot of viruses. These are now carried back through the axon to the skin, and will generate these shalpuchiot, I don't know. So you'll have like red skin on this area, but it will kind of mark only like one part of your body, very, very clear band. And this band is basically this dermatome, because it's kind of transfected one ganglion. Yes? Do we use it?
Yeah.
So herpes viruses they were very useful at the beginning for retro-labeling, yes. People were using them. Still, they're still using them, but we have now, people were using them because of this property that they can go back, still being used, but we have much better ones now.
But I just meant going back and then going forth. Is it a property of this specific type of herpes?
I'm not sure. I'm not sure. I mean, if only herpes virus are doing them. Okay. There's certainly one of there are different ones that can do this retro transport. Not all of them can. Okay. So some viruses, if you transfect the nerve endings, they will not don't have the machinery to go back with the axon. Some of them do. Okay. Okay. כן, בחגורה. Yeah, exactly. What else is there? Okay, so the last thing I want to talk to you about today is the fact that I told you that they have like these different pathways where they go through the brainstem. What is important to realize is that they actually keep the identity in several ways, all the way to the cortex. So, for example, the inputs that are going from lower part of the spinal cord. So here, these are example of 3 axons that are going into the spinal cord at these locations. Okay. Sorry, what you see here are kind of different cuts through the spinal cord. So every cut here is a cut here. Okay, so the red axon are going here, the blue one here, and the green one here, and then you go already to the area of the medulla. So this is the next one here, And you see that they kind of maintain. So for example, the red one, they're going up in the spinal cord and then make a synapse and cross to the other side. Okay, and some of them are doing this crossing here. Some of them are going directly already to the side that they continue. They go to the thalamus and then they project differently to different locations on the somatosensory area. And what would that make? So what's the difference between these ones and these ones and these ones? No, so say we are only looking now here on sensory, Meissner corpuscle, I don't know.
Where in the body is the information?
Right, so these would come from the legs. Okay, this would come from the hand. This would come from the face. So they would map differently on the cortex, and this is basically, you can see it here, and this is what makes this homunculus that you see on the brain. Okay, so the legs would be projected over, here, and the tongue and the face would be projected over here. Okay, so this is what makes a homunculus. Within every area, we have also sub-mappings that I'll show you next time that talked about which type of receptors are doing that, but we'll do that in the next class. Okay, on Thursday. So we can stop now and continue on Thursday. כן. לא, זה לא שאני לא מאמין באבולוציה, אני פשוט לא מאמין בה, אני חושב שציונים אבולוציוניים נרקים בצורה יותר מדי אוף הלט כזה, ואנשים בצלילים, הרבה פעמים אנשים נוטים להסביר למה קופים יודעים לעשות איזשהו משהו, למרות שקצת העלינו שנתיים באבולוציה לפתח אותם. לא יודע, אני חושב שהטיעונים האלה לא נחמדים, אפשר לדבר עליהם, אבל לא, אני חושב שאבולוציה זה פשוט משהו נורא נורא מסובך, שאנחנו מבינים ממש כאילו רק את הקטע שלו, וגם נורא קשה להוכיח כל דבר בצורה שהיא קודלית. בשבילי אחת הדוגמאות הכי קלאסיות זה הדוגמה הזאת של השיר של הסיפורים. אני חושבת בזה, ואת אומרת, הסיפור הזאת משפיעה כל השלושה חודשים הראשונים של החיים שלו וללמוד שיר. והשיר הזה, נכון, הוא יעזור לך כנראה למצוא בזום, אבל אין לו שום קורלציה עם כמה הסיפור הזה צריך לבוא בתור אבא. זה לא אומר כמה טוב הוא בונה כן, זה לא אומר כמה הוא חזק בשביל להגן עליהם, זה לא אומר כמה טוב הוא בלמצוא אוכל. אם כבר לדעת קורלציה לזה בגלל שהוא משקיע את זה טוב בלימוד של השיר, אז זה לא נשמע כל כך משכנע. ואז אנשים באים עם הרעיון הזה של עיקרון ההכבדה, שהוא נחמד, אבל אני לא יודע, אני חושב שיש בו משהו, אבל אני לא חושב שהוא כל כך אותי, למשל, לא לגמרי משכנע. ואני חושב שיש הסברים אלטרנטיביים שמראים, בדוגמה הזאת, בוא נניח מצב שבו איפשהו באבולוציה מאיזושהי סיבה, היה איזשהו מקרה שהציפור השמיע קול, וזה גרם אצל הנקבה, במוח של הנקבה, לא יודע מה, זה שיפור של דופמין. היא עכשיו כאילו רואה את השיר שלו בתור ריבורד, ואז היא מגיבה אליו, ובעצם עכשיו היה, לא יודע איך לקרוא לזה, מחטף כזה של האבולוציה, על מין מנגנון שבכלל לא היה אמור, זאת אומרת, זה קרה במקרה, ובגלל שלא יש מוח, ולא יש דופמין, והוא שם, נכנסנו לאיזשהו מעגל של פידבק חיובי שאי אפשר לצאת ממנו. ומה זה קשור זאת אומרת, האבולוציה יכולה להיות גם סנסיטיב לדברים שהם בכלל לא רלוונטיים, והיא עדיין ממשיכה לפעול בתוך הדבר הזה. כאילו, אני חושב שהטיעונים האלה צריכים נורא לדעת איתם, כי הם נורא פשטניים, והם לא נותנים את המושגות של כל במיוחד כי יש מוח מעורב בכל אלה שלהם. אני חושב מה המנגנון
זה אולי הכי חזקה
של הפיטנס הוא הרבה יותר מסובך, כולל דברים שלאו דווקא קשורים לסרווייבל.
באוגוסט 47
אני משתדל להשתמש בטיעונים האלה.
אם זה לדבר היה מצא ראשונה של הלגישה, זה לא קרה בשנה בשבילה יום אחד.
זה רק כזה שיש מחשבה שהנוירונים במהלך של השיר, כשהציפור שרה זה איזשהו כמו סינפייל אבל לא הבנתי לגמרי, כאילו בסינפייל של הרעיון היה איזה מיני קואינסידנס דיטקשן ברמה חצי לא הבנתי איך זה קשור כאן, כאילו זה נראה משהו מאוד סדרתי, זה לא איזה משהו שנקפיים לאורך או כאילו שכזה יש, אתה מזהה כמה אג'ז ואז זה משהו יותר גדול
האג'ז פה
צריך לשים את המאמר הזה, את המאמר שלך,
אבל כן, אמא שלו, זה הגיע דרך אמא שלו ולפחות, אבא שלו לא היה אמא שלו ואז דברים שהוא זה, אבל זה יש טיעון מאוד חזק, כאילו אתה יכול לקרוא את התביעות, אתה רואה כחול, אתה לא, מה המשמעויות של מה כחול, מה המשמעות של האם אתה מעיין במשהו ולא מקיים אותו יש שם תיאורים כאן ממש כואבים של שני הצדדים, יש גם את התיעודים של מה קרה עם שלושה האלה בכלא שנסו להורג אגב, אני חושב שהמטרות מאוד נפסים כמו בנקודת של ניסים השתתפו בכניסה לכלא עכו שגם כן הייתה שאחד גדול ואז עכשיו כן שהוא בלתי כל כך להבין הכלא של אז זה היה מעצמה כמו שפוליטה שהיה מעצמה עתיק והם הוסיפו שעושים אפשר וככה ואז חסם וכן, כל הסיפור פה הוא סיפור מאוד 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 מרשים כאילו, בכל הכיוונים אבל קיצור, זה לא סיפור, זה פשוט אני חושב שפחות המבנים על הזמן זה מספרים שהוא צעד מהכלא אז אתה לא יכול להבין על סיים יותר יכול שפחות סוף אבל כאילו זה בא לפני זה אבל
זה מספר על התהליך זה יותר בקטע אישי או בקטע טוב
מהמצב שאתה לא מדבר כאילו אז יש פה מספרים שהוא כותב למשפחה וחברים במקרה של ויש פה אבל גם מספרים שהוא כאילו כותב לתינוק או בייבי ומוצר כאלה אנשים כאלה שהם קצת כאילו פוליטיים אבל זה מאוד מאוד ציני כאילו לקרוא את כלומר כן יש החבר'ה האלה הם היו תגיד שזה הפרקטות השקלים אבל ממש כאילו שום דבר זה ממש שום דבר שבודקים את המיצג שום דבר
הרעיון הוא כאילו שאתה מודד אינטרסט בתוך הנוירון הזה שהחסר מרתיק ואתה רואה שבמשך כל השיר כאילו לא קורה כאילו שום דבר אבל בזמן שזה מגיע כאילו יש איזה גבולת כזה של הרבה מאוד ואז הוא יורד ואז הוא חוזר זה כאילו הסיבה שחושבים שזה בעצם יש איזו קבוצה שמפעילה קבוצה
והאינפוטים והנוירון הזה זה האוטפוטים של הדבר הזה זה מה שעובר לנוירון הבא
אז הנוירונים האלה עושים שני דברים אחד יש להם קשרים בתוך הלגיטי והדבר שבאמת יותר מעניין שזה חשוב לסינפייל והדבר השני שמעניין זה שבעצם הפרוג'קשן של הנוירונים, איך הם בוחרים את הנוירונים האלה? זה מאמר די משוגע, אבל הם עושים, הם כאילו עושים ציפור מרדימים אותם. הם בונים איזשהו מכשיר שיושב את הדבר הזה, יושב על הראש של הציפור.
זהו, זה היה נראה לי גם, אבל
כי הציפורים האלה לא שרות, לא יכולות להסתובב חופשי. אז יש להם מנוע קטן שמחזיק את האלקטרודה ואיתו הם יכולים להתקדם עד שהם תופסים נוירון חוף. במקביל, החיה כרגע מוקדמת, אז הם יכולים לשים עוד שתי אלקטרודות בשני הגרעינים האלה. זה באזל גנגליה של הציפור, וזה RA שזה כמו המוטור קורטקס של הציפור. ואז הם עושים כאן גירוי שנקרא אנטי-דרומיק סטימוליישן. זאת אומרת, הם מזריקים פה זרם, גורמים לעצמם להעביר פוטנציאל פעולה בכיוון ההפוך, ומודדים בנוירון הזה כדי לדעת, אם הוא פרוג'קטינג לכאן או פרוג'קטינג מכאן. כן, יש כאן 3 סוגים של תאים, כאלה שהולכים לפה, כאלה שהולכים לפה וכאלה שנשארים בפנים.
והם רוצים לשלוט את האדומים.
הם רוצים את האדומים. אחרי שהם תופסים את האדומים, הם מביאים את הציפור ונותנים לה לשיר, ואז הם מודדים מהנוירון הזה שהציפור שם. אז מה שאפשר להראות זה שהגרעין הזה, כשאתה עושה לו next stimulation, כל פעם שאתה עושה stimulation פה אתה מקבל חלק מאיזשהו סילבוס. אז הרעיון הוא שבעצם הנוירונים האלה, זה מה פה בזווית
זה בלתי אפשרי לבצע עבודות.
אז מתחרים נגד מישהו כאילו?
אתה בקבוצה מתחרה נגד מישהו וכולם מפסידים למישהו הזה.
זה מישהו כאילו מורכב בגרוף?
משהו רוצה להראות זה שכאילו יש כאן ב-HVC יש כאילו, זה הסיפור, הקבוצה הזאת יורדת, כל אחת מהן מפעילה איזושהי קבוצה כאן ב-RA ובעצם כשאחד יורד זה מפעיל את זה ואת זה וזה יוצר סילבל אחד, כששתיים יורד זה מפעילה את זה ואת זה ויוצר סילבל אחד וככה כאילו אתה יוצר את הסילבל לאורך הסונג ובעצם הסונג פה, אם אתה מודד ב-HVC אתה רואה את הקבוצה הזאת, אחרי את הקבוצה הזאת, אחרי את הקבוצה הזאת, את הקבוצה הזאת זה ה-Same Fire Chain וכאן אתה רואה שהקבוצה הזאת וזאת פועלות ביחד, זאת וזאת פועלות ביחד, זאת וזאת פועלות ביחד, כל אחד מהם יוצר סילבל אחר.
מייחד את החיבוריות הזאת.
כן. לא, זה בעצם הקונסקוונס, אבל כשזה פועל אתה מקבל אלפא, זה כאילו סילבל אחד, כשאלה פועלים אתה מקבל סילבל אחר, ואז זה אומר עכשיו תעשה אלפא, זה אומר עכשיו תעשה בטא, זה אומר תעשה גמא, וככה אתה מייצר את השיר. ויש להם עוד עדות מאוד יפה לזה, שמה שהם עושים זה זה המאמר שאני עושה קודם, פה, זה לא מאמר מאוד יפה, ומה שהם עושים כאן זה טרנספורט בדלטה, אבל מה שהם עושים כאן זה הם מקררים, הם עושים מכשיר שזה מתח, הם יכולים למצוא בטמפרטורה שלו הם יכולים לקרר את ה-HVC כי ממש נמצא קרוב לקצה של הגולגולת פה ואז זה השיר וכשהם מקררים את האזור אז השיר בעצם הולך ומתארך ובעצם הרעיון הוא שככל שאתה הופך את זה ליותר קר אז הקונדקשן ולוסיטי יהיה יותר איטי ולכן בעצם הדיליי בין גרופ לגרופ יהיה יותר איטי
והעברות נשמרות אבל...
כן, אבל הן נלתחות ומה שהם מראים כאן זה שאתה הופך את השיר הזה ואתה שהוא 38% יותר איטי אתה לוקח את הקונטרול ואתה פשוט עושה לו סטרץ' בזמן בלי שום מניפולציה אחרת ואתה מקבל בעצם את אותו שיר אז זה ממש כאילו מעכב את זה כאילו זמן ואם אתה עושה את זה בעלי אז אתה לא מקבל זאת אומרת זה כאילו זה מניפולציה על הזמן יותר מזה יש לו איפה זה נמצא? שיש כאן את הכל כן אתה יכול לעשות את הקירור הזה ב-HVC ימין ו-HVC שמאל יש לו צדדים אז אם אתה מקרר אחד אז אתה מעריך חלק מהשיר ואתה מעריך את השני אתה מעריך חלק אחר וביחד אתה מקבל את האפקט שראית למעלה זאת אומרת זה מין זה כאילו השיר גם כנראה קופץ בין צד ימין לצד שמאל וחוזר לצד ימין זה מאוד
You told about resolution and the dimension of the reflective field, saying that, for example, in the back, the reflective field is very large and the resolution is not good, but isn't it compensated by a larger amount of Pacinian corpuscle, for example, in the back to, even if the receptive field is big, to have to compensate and have a resolution that is comparable to...
I don't think so. I think the resolution in the back is indeed less good than the other.
Yes.
Of course, than the
yes, but well, I mean, is the resolution is not the same thing as the width of the reflective field, no?
Because
No, of course. I mean,
yes.
you would. I think if you do that to someone and ask him, do I touch in the same place? It would be harder to tell. So obviously, if I touch here and here, you know that I do that. If I take this distance on the back, you might not know that. But if you would kind of, do something like, something that includes movement or something like that, then the resolution would be better because you can integrate in time and use other receptors, so you can improve the resolution. It's kind of hyper acuity like you have in the visual system.
Okay, because the receptive fields overlap, I guess, between.
To some extent, yes.
Okay.
Yes.
So we can rely on several corpuscles to convey the.
Yes, true. But still, I think the resolution, obviously, on the back is less good than on the back.
Yes.
But you can do, you can do much better. For example, the edge here, right? You can make, you can show cases. I think one of the best cases is if you take a sharp knife, okay, and you pass it on your nail. Just do that. Okay? You can tell if it's completely smooth or it has some dent. And this dent could be they could be microscopic, they could be way smaller than a millimeter. Although you don't have a resolution, suddenly on the nail you don't have a very good resolution. Right? But because when you move it, you have like some kind of vibration, you can see it move, and then you have something, and you immediately notice something like that. So you can have, a lot of hyperacuity in this. I think the best resolution somehow is in the tongue and in the eye only. These are the two places which are most, for some, I think in the eye just because to protect it, and in the tongue also because I think it's interesting, but not so many people study that. The texture of food is very important, and we tend to think that everything is smell and taste. But texture is actually very important. You have a lot of texture.
And even for speaking, we need to have a proprioception.
Sure. But this is not touch, so you have like proprioception. It's also very, very dense.